Welcome, everyone. So we've just heard that we have 50 attendees, which is amazing for a webinar. Thank you so much, everyone, for making the time this evening. This is a webinar, a first really sort of that we're hosting a company that is talking about an ASO for Syngap 1. So we're really excited to welcome CAMP4. Before we hear from them, we at SRF have a very general introduction to clinical trials. Just a couple slides that we want to take you through that Lauren, thank you so much, is finding. We've included a QR code with a glossary of terms. A lot of the terminology is medical and things that we are not using in everyday language. If any of the terms stump you and you're thinking sort of, I just want to check another time and see what that is, go ahead and use that QR code. If anything comes up that's not in that list, please let us know and we'll make sure to add it with a sort of short explanation of the terms. So that's great. Sorry, Lauren. Maybe just go to the third. Yeah. So again, this is just a really short introduction from the Syngap Research Fund on clinical trial development. So first, really important to note that all new therapies, drugs, need to be approved by the relevant government agencies. In Europe, that is the EMA. In the US, it's the FDA. Australia, they have their own. So every region will have their own sort of medical agency that will need to go through the process of approval. Today, we really use randomized placebo-controlled trials. So what that means is that when you enter a clinical trial, you are not aware if you're getting the actual drug or if you're getting a placebo. And in fact, your doctors don't even know what they're giving you either. This is effectively the gold standard. And what it does is it prevents the placebo effect. So it prevents any milestone that your Syngapian might hit that you think can be attributed to a positive on a new drug, or any negative. They have like a really bad day. You're thinking, oh my goodness, like, this might be a negative side effect. So this way, if everyone's not in the know, they can actually measure through data, present that to the government agencies, and it is a much more rigorous process. Before any clinical trial begins, the medicines are tested extensively for safety. They're tested in monkeys, mice, cell lines, as we'll sort of hear today. And this will sort of effectively show, are these drugs effective? Are they actually improving? Having improvements in outcomes that we want to see. And is the drug working the way they think the drug is working? So is it is the mechanism of the drug, in our case, is it actually making more Syngap protein? So those are sort of a few things to consider. Ryan, can you hit next slide, please? Yeah, thank you. So here's a general overview of a timeline for clinical trial development. The top half of the slide would show a sort of typical process. In cases of rare disease, this is sort of more compressed, shortened. Effectively, we just don't have the patient numbers to support 3 phases of 100 plus patients. So typically what would happen is phase 1, 2 is put together. It would be tested in patients. Multiple dose levels would be tested. And this is looking at the safety of the drug. It's looking at the dosage and the efficacy of the drug. Phase 2, 3 would be actually to confirm the findings in phase 1, 2. And then actually in our case, we might have a chance to have to go for approval in that phase as well. Whereas typically sort of in a normal trial, as you can see above, the approval phase is right at the end of that. So one other thing that I just want to pause on is the open label extension. It's really important, I think, for us and for people today considering future clinical trials. So open label extension allows patients that are in the earlier phases to continue on treatment during phase 2, 3. So if you are given a drug in phase 1 and you are keen to continue on that drug, that would be supported throughout the whole clinical trial process. And if you are given a placebo in phase 1, 2, you will have access to the actual drug then as well in the later phases. So that's a really important consideration for everyone to keep in mind. So why participate in a clinical trial? The short end of it is the absolute fastest way to get access to disease-modifying therapies. We, as a community, are very, very fortunate that we have so many companies that are looking at SYNGAP1 and that are developing treatments for SYNGAP1. There are many, many rare disease groups that are a lot further behind than we are in terms of science and having sort of company interest. So we are very, very lucky to be at this phase where we are right now. So we should definitely take advantage of that as well. ASOs, so specifically what we're speaking about today, very unlike gene therapies, they are not a one-time and done drug. It is a drug that is consumed by the body and then needs to be redosed. So this is actually sort of, as we see it now, sort of can be seen as a very positive way. It means that you can start at a low dosage. It means that also if there were to be any sort of very negative side effects, that drug would then phase out of the body. So it's something that can be adjusted according to how things go. So that's really important to know about ASOs. I've mentioned that was sort of very, very lucky to be diagnosed as well, as we sort of have this great community that is looking still for many, many undiagnosed Syngapians. But for those of us who do have a diagnosis, please know how fortunate we are that you know what is causing the problems that you're seeing and the symptoms that you're seeing in your child, and that we now actually are looking at potentially having an opportunity to fix that. Lauren, can you hit the next slide for me? Thank you. So ASOs are in trials for a number of other DEEs. So we're certainly not the first. We would love to be added to this list as soon as possible. So we've listed here a number of trials that are on and which phases they're in. So Stoke and Praxis are names that many of us will be familiar with, as they are also working on SYNGAP1. The other ones, so Angelman has a number of ASO trials that are ongoing now, as well as RET. You can click onto all of these links, and then that will take you to the information on those trials as well. So that might be something you want to do later. So this is a last sort of hygiene housekeeping slide that we just wanted to provide everyone with some context when it comes to clinical trials and when it comes to us as a patient organization giving you information, but also when we speak to companies that we need to be mindful of the information. Some information will be limited at this time. So for example, CAMP4, they are a listed company. They have to consider their financial regulators. They have to consider the medical agencies. They have to consider the phase that they are at now and where they are. So when they're giving us information, that is a consideration they need to keep in the back of their minds. For the patient advocacy groups in Europe, there are so many. We're really, really thankful to the Syngap Global Network that helped us put together the 50+ participants of today's webinar to promote this and to spread this information throughout Europe. All of those local Syngap organizations, they need to consider their patients. They need to consider the multiple companies. And so we need to be as neutral as possible And so please bear that in mind when we are giving you information. It is there as information and not advice. And finally, leaders, you know, we all of us have Syngapians of our own. We all have to consider whether a clinical trial is right for our child, our family, and that's a decision that we need to be taking within our family, with our spouses, with our children. And so it's not something that everyone's going to be public about. And so please be respectful. This is a personal decision for everyone. If you do get to participate in a clinical trial, then there will probably be a request or something slightly more stronger than a request that you don't share public information about the trial when it's ongoing. The reason is you just don't want to influence other patients and sort of see whether it's a positive effect or a negative effect. It will sort of make people think like, oh my goodness, I need to be on the lookout for this, or I need to be on the lookout for that. We really want to sort of make sure that everyone is coming to the information and the conclusions that are based on themselves and how their child is doing in that trial. So just a little reminder there for everyone. So that is it for us. Thank you so much. And we're going to introduce CAMP4 now. We're really, really thankful for your time. And we're really looking forward to hearing more about your ASO for Syngap 1. Welcome.
Great. Thanks, Katrien. Really appreciate it. Bear with me 1 second here to make sure everyone can see the slides. Those come through in full screen?
Yeah.
Yeah, great. Excellent. Well, thank you very much, Katrien, for the very warm welcome and introduction. And thanks to the broader, the SRF EU community. I will say my colleagues and I were just sort of texting offline and just kind of floored at the turnout, right? Really impressed at the number of people that are turning out. So really appreciate everybody tuning in to both learn more about our program, but also to educate us, right? This is a mutual collaboration here, and in each one of these webinars that we've done, we've learned something from the community as well. I will start with just a quick anecdote. SRF has been a great collaborator, a great partner from the early days of our Syngap work. We first met Mike Graglia at AES in 2022 in Nashville, where he saw a very early poster that we had put together that one of our scientists presented. Mike was very eager at the front of the line and said, "Hey, you guys, we have iPSCs. You need to be working with them. You need to be testing them." And then we were off to the races. And that was really kind of a pivotal moment for us, because it's one of the things that catalyzed this going from a research project to a full-blown program and being put on a clear path to the clinic, where we organize a lot more resource around it. So we've had a great collaboration with SRF. We're thrilled to meet the EU community. And we look forward to a really fruitful and productive partnership going forward. So my colleagues and I thank you, Katrien, for giving the disclaimer around we're a publicly traded company. We're going to make forward-looking statements today, so please bear with us. I'd like to start just with to give everybody some context for who is CAMP4. Our mission is we're really pioneering a new class of medicines to increase gene expression. And more specifically, we're pioneering the discovery and development of regulatory RNA targeting antisense oligonucleotide, or ASO-based therapies to upregulate gene expression and restore healthy protein levels. Now that is, there's a lot of jargon in there and it's a mouthful, but I want, our whole point in being here today is to break that down, simplify it, put it in terms that are really understandable and digestible, so you understand how our technology works and why we're really excited about it. Fundamentally, CAMP4's special sauce is our understanding of these regulatory RNAs that our medicines target and the role that they play in gene expression. We're going to talk a lot about that today. But fundamentally, our goal is to increase Syngap expression or make more Syngap protein. Yuri, Dan, and I are here today representing roughly 45 employees. We're all sitting in the office today. We're headquartered in Kendall Square in Cambridge, Massachusetts. And the founding science behind our company comes from the Whitehead Institute, Boston Children's Hospital, and Harvard Medical School. So what is an ASO, and how might it be applied in the context of Syngap1? So ASO stands for antisense oligonucleotide. And there's lots of terms that are used to describe ASOs, right? We use the acronym ASO. People spell out ASO. They talk about oligos or nucleic acid therapies. These are all the same things. And what these are small nucleic acid molecules that can be engineered to target specific sequences of RNA. And ASOs can fine-tune genetic instructions, helping regulate production of a specific protein. In our case, we're developing an ASO that aims to increase expression of the SYNGAP1 gene to near normal levels, improve how neurons transmit signals, and alleviate the symptoms of SYNGAP1 patients. I want to call out that we're using standard ASO chemistry like that of Spinraza, which is an ASO that's been approved for spinal muscular atrophy, or SMA, from Biogen. And I call this out because the chemical backbone of our medicine is nothing new. It's been tried and tested. So we like to say that we're pointing a known tool at a new target. And again, back to the concept of special sauce, you're going to hear over and over that our understanding of these regulatory RNAs as the target for our ASOs is really critical and is what's kind of unique about our understanding and our approach to Syngap. Katrien did a really nice job contextualizing ASOs relative to some of the other therapies or types of medicines that are out there. But we thought it'd be helpful to situate among the types of medicines that come up more often in the context of treating CNS disorders. So we've listed small molecules, gene therapy, and then ASOs from left to right here. And small molecules are the medicines that most of us are probably familiar with. They're small compounds or they're typically pills. They're taken orally. So you take them by mouth, they travel to the gut, they're absorbed into the bloodstream and distributed throughout the body, right? A lot of them go to the liver, get processed there, and then they get excreted. And some of it gets to the intended targets or target tissues that you want. But you have to, because they're excreted, you have to take them repeatedly, typically daily. And the role that they play is they bind receptors and proteins and elicit some sort of impact on signaling pathways in the body to have a therapeutic effect. And if you move to the next column, gene therapy, this was touched on by Katrien up front, and this is sort of taking a synthetic gene and packaging it in a way that it can be delivered. So typically they use a virus or a viral vector to deliver the gene therapy. And this approach is one whereby the gene therapy is shuttled directly to the brain. It's administered one time, or the intention is that it's administered one time. And this is an important point because our bodies our immune systems are very good at dealing with viruses. And so you've kind of got one shot to deliver the virus and deliver the synthetic gene. And then our body knows how to tackle it if it were to be given a subsequent time. So this is more of a permanent approach, and one where the dose can't be adjusted or washed out. And then finally, on the right-hand side is the ASO technology that we've talked about. And again, these are short genetic sequences that are very good at very specifically binding RNA sequences. These are delivered via intrathecal or IT injection. And we're going to spend some time talking about intrathecal delivery. But fundamentally, we want our ASO medicines to reach the cells that matter in Syngap. So it's injected into the spinal cord and travels directly to the brain. Again, this is the intrathecal injection. ASOs that have been used for other CNS disorders are typically administered monthly to quarterly. We've talked about the fact that they bind specific RNA sequences, but one of the really unique features of ASOs is that they can be dose adjusted. They can even be stopped. And in some cases, you can sort of titrate up the dose to have as you're administering to elicit the intended effects that you'd like on patients. ASOs are nothing new, and ASOs to treat CNS disorders are nothing new either. They've been around for a while, and the benefits of ASOs have been shown with multiple approved medicines. We split this into sort of two perspectives on this slide, but on the left-hand side, you see what's known as the sort of the central dogma of molecular biology, whereby DNA makes RNA, which makes protein. And ASOs can target pre-mRNA or mRNA in a very specific way to elicit effects on gene expression and protein production. And you can see our ASO, which again acts at the level of the regulatory RNAs, which is high up in the progression, right, at the transition from DNA to pre-mRNA, and has the effect of increasing Syngap1 expression. And the table on the right-hand side lists the approved antisense oligonucleotides or ASO therapies that have been out there for other diseases and other different types of delivery to target different tissues. And you can see that the other approved ASOs for CNS diseases have also been delivered by intrathecal delivery. And in fact, there's more than 14,000 patients that have been treated with Biogen's Spinraza for spinal muscular atrophy, which is another IT-delivered ASO. So I'm going to pass it over to my colleague Yuri to talk a little bit more about intrathecal delivery and our mechanism of action.
Great. Thanks, Caleb. And it's a real pleasure to be here. My name's Yuri. I'm the Chief Medical Officer at CAMP4, and I've also had the pleasure of meeting a number of SRF families and your Syngap family members. So maybe just to pick off where Caleb left off, I think it's important that we all consider what is oftentimes the optimal or best approach to deliver the potential medication to the organ that we need to treat. And as Caleb mentioned, that there have been a number of ASOs that have been utilized to deliver diseases of the brain. And in particular, Spinraza is a great example where now thousands and thousands of patients have actually been treated. And so intrathecal, or IT, you'll sometimes hear called administration of ASOs, allows us to then reach the appropriate or target cells in the brain. Sometimes this is also known as lumbar puncture or spinal tap. And this way of delivering the medicine allows the medicine to travel through what's called the CSF, or cerebral spinal fluid, to get it to the parts of the brain where that CSF bathes. And what's also important is while it delivers the ASO there with the appropriate dose, it also reduces the amount that might get to other organs that we do not need to get to. So for example, depending on the disease, we may not want to have to deliver, or we want to avoid getting high doses to, say, the heart or the liver or the kidneys. And so by delivering it to the appropriate place, that increases the potential benefit and it reduces potential risks associated with it. A challenge of giving ASOs into the bloodstream, if we want to get them to the brain, is that there's the blood-brain barrier. And this is a strong protective effect for our brains. It prevents many chemicals from getting to our brain, and ASOs are no different. And so that's why delivering it intrathecally is important. I do want to point out that intrathecal administration for many types of patients is done under some form of sedation. And so that is quite common. And given the thousands and thousands of patients who've been treated, ASO administration and intrathecal route is quite common. So many neurologists and pediatric neurologists and epilepsy specialists are very accustomed to this and have lots of experience of doing this safely. We go to the next slide. So because of its effectiveness, there are a number of ASOs that are also in the clinic. Katrien mentioned several of these. So these are additional clinical trials focused on other diseases of the brain where the ASO is actually being delivered intrathecally. So there's Angelman syndrome, Dravet, SCN2A for Praxis. And so these are all examples of, because of the effectiveness, this is a pathway that really balances the benefit with the risk. And if we go to the next slide here, I think the thing that is important is, as we think about the approach, we want to match both the route of administration, but also the type of medicine or the technology that is part of the medicine to the disease in the most appropriate way. And the background around, as Caleb mentioned, our technology that I'll walk through a little bit more, and then Dan will go over some of our data, really lends itself to particularly benefit haploinsufficient diseases. And haploinsufficient diseases, of which Syngap is one, is where you have one copy that does not have a mutation and another copy that has a mutation. So you can also simplify this to one good, one bad copy. And in healthy individuals like ourselves on this, us as individuals have two healthy copies, one from each of our parents. And so we produce a normal amount of Syngap protein. However, a Syngap individual, on the other hand, would have a mutated copy and either makes no or little protein from the affected copy. And so they only make roughly 50% of protein. And this is what results in the condition that they live with. However, if we go to the next slide, what you'll see here is that the kind of central dogma of biology that Caleb walked through, so DNA makes RNA, makes protein. There are multiple ways where we could try and get more effective protein. And so to continue to use the example of Spinraza, because it's a good one, it functions in between what is called pre-mRNA and mRNA and helps convert more of that pre-mRNA to mRNA that will then make protein. The approach we're using at CAMP4 is actually to target at the level of DNA. And so our focus is to help the healthy copy of Syngap make more. So we are actually just harnessing that already healthy allele and having it to make up for the loss of Syngap protein that comes from the mutated or bad copy. And the next slide here will kind of show how we do that. So we have identified these regulatory RNAs, and these are components or machinery that instruct the gene as to how much to make in terms of the mRNA and then ultimately the protein. And you can see that in the cartoon in the bubble. So there's machinery that comes around the gene, and there are sometimes what are called activators. They tell the gene to make more. There are also repressors, which would tell the gene to make less. And what we identified was, in fact, that there was a repressor associated with the gene. And you can see that in the little pink diagram there. And we learned that if we target that with our antisense oligonucleotide and we have a sequence that binds to that repressor, it actually causes the repressor to come off the gene. And so that is equivalent to like taking your foot off the brake in a car and you get increased protein. And so what we'll see in the data that Dan's going to show us now momentarily is that we've been able, by targeting the healthy allele and causing more protein to be produced, that we can actually take 50% protein and bring that to near normal levels.
All right, thank you, Yuri, for that background. I'm excited to tell you about our preclinical data, our in vitro and in vivo data with our candidate oligonucleotide to increase Syngap expression. This has been a lot of work over the last couple of years by our team. And in addition, just to give you an overall view, in addition to Syngap, which we're approaching toxicology studies, we are a clinical stage company. We are using this technology to explore urea cycle disorders, which is a rare disease, rare liver disease, and this is currently in a phase 1 trial. We also have another, have other interests in other haploinsufficiencies, as well as in GBA1 Parkinson's disease. So these are all, what's coming up behind Syngap. But for the rest of this talk, I'll tell you about our work in Syngap. So I'd like to start off by telling you about some data that we have in patient iPS-derived neurons. And what are these cells? You may have heard of iPSCs. Essentially, these are cells that are generated from skin cells or fibroblasts from an individual. So you can do a skin biopsy. You can take these fibroblast cells and treat them with a few genetic tricks to turn them into what we call induced pluripotent stem cells. And then these iPSCs, we can then differentiate into different cell types. In our case, we want to turn them into neurons. And the advantage here is we can take cells that are essentially coming from a Syngap patient and compare them to a familial control, in this case, a sibling. And what we see in the data on the right, this is looking at Syngap mRNA levels in a Syngap patient in gray, and then in the orange color, a familial control. Again, this is a sibling. So you can see that the patient with this mutation has a 50% reduction in Syngap level. And when we treat these neurons with our oligonucleotide, we see complete recovery of Syngap levels back to that of wild type. So this nicely shows that we're able to identify this regulatory RNA, and it's working in these patient iPS cells to derive neurons to regulate Syngap expression. And by targeting this with our oligonucleotide, we're increasing gene expression back to wild type levels. Next slide. So it's really important that we are able to translate activity of our oligonucleotide into an in vivo setting. So in the context of brain tissue where neurons are highly connected and functioning to support the life of these animals. So we were able to use a very clever mouse model that came from Ben Prosser's group at UPenn. This is a humanized mouse in which human Syngap, including the promoter and all the regulatory elements that are making the regulatory RNAs that we're targeting, have replaced the mouse Syngap gene. So the homozygous mice, that means it has 2 copies of human Syngap, no mouse copies, and then the haploinsufficient mice have a single copy of human Syngap, and again, no mouse copies. So this is nicely recapitulating a wild type versus haploinsufficient state. And you can see, comparing the orange to the gray bars, that in the haploinsufficient Syngap mice, there's a 50 to 60% lower level of Syngap protein. So the mutation that's in this gene is resulting in less protein being made. And importantly, we can take our oligonucleotide, the same one that's targeting the human sequence, and administer it to these mice directly into the ventricles in the brain, so into the CSF within the brain. And the ASO distributes to the brain tissue, gets into neurons, and is actually increasing Syngap protein levels by about 2-fold. You can compare to the CSF group, which is just the vehicle artificial CSF, and you can see a nice 2-fold increase in Syngap protein levels. Near wild type levels are achieved. So this is really nice data demonstrating that the effects that we saw of the oligonucleotide in vitro in cells in a dish, even though they were human origin, is able to translate into increasing Syngap in the context of an intact brain in a humanized Syngap mouse. Next slide. So importantly, we can take this mouse and then start looking at Syngap-dependent behaviors and the effects of our oligonucleotides on that. So think of this as a mini clinical trial, if you will. So the mouse Syngap heterozygous mice have been characterized in the literature and have been shown to have certain phenotypes associated with motor function, with cognition and memory, as well as with hyperactivity and anxiety. And those are the tests that we ran in this particular study. So we characterized the haploinsufficient humanized Syngap mouse. And you can see, comparing, again, the gray bars, which is a homozygous mice, to the yellow, which is the heterozygous mice with the mutation. You can see that there are differences in all of these measures, so that there's a Syngap-dependent phenotype. And in all cases, when we administer our oligonucleotide, you can see that there's a restoration of wild-type behaviors or activity in the mice receiving the oligonucleotide that's increasing Syngap expression. And then when you look at the right graphs, this is actually looking at the Syngap protein in the mice that are being assessed in this behavioral study. And you can see that there's a significant and robust increase in Syngap protein in cortex, as well as hippocampus, which are just 2 general brain regions that are important for manifestation of a number of the phenotypes associated with Syngap disorder. So again, nicely showing from the prior slide that we're increasing Syngap. Here, there's actually a phenotype dependent on that reduction in Syngap levels, and our ASO is reversing those phenotypes. Next slide. So this is, we're extremely excited about this data. It really suggests that we're intervening and able to have an effect on Syngap that's meaningful in these animals. You'll notice that when I described that study, we were administering the oligonucleotide directly into the brains of these mice. That is for technical reasons. Mice, it's hard to deliver intrathecally. But IT administration is the route that is going to be used in our clinical study. So it's important that we can show in non-human primates that administration of our oligo into the lumbar spinal cord region, and that ASO can travel up through the CSF into the brain and distribute to the tissue and increase Syngap expression. So we ran a study in which we had 3 different dose levels, low through high. We administered the ASO every 2 weeks for 3 doses. And then we sacrificed the animals and looked at the brain tissue, which is just an important part of being able to assess the impact on Syngap levels, and looked at ASO concentrations as well as protein levels. And the key takeaways from this study are, 1, that the ASO was very well tolerated. There were no clinical signs or symptoms associated with the administration of our oligonucleotide candidate. And importantly, in the data that I'll show you in the coming 2 slides, is that the ASO got to all the relevant brain regions in a very nice dose-dependent way. And also the concentrations of ASO that we were able to achieve were sufficient to increase Syngap levels to a significant degree that we feel could be beneficial. So now I'll take you through that data. So here we're looking at some different cortical regions within the brain, sort of the front or the sides or the back, as well as motor cortex, and then hippocampus, which is important for learning and memory. And you can appreciate that 2 things. 1 is that we're getting fairly even concentrations of ASO in all these different brain regions. And then you can also see that it's very nice dose dependent, meaning if we give 3 times more ASO, we get 3 times more ASO in the tissue. So it's very important that we get this dose dependent and dose linear effect, which helps us think about future clinical study design in terms of the dose levels we might use. Next slide. And critically, and what is absolutely essential for this to be a drug, is that the ASO that's getting into the brain tissue, into all these regions that we feel are important based on disease phenotypes, are increasing Syngap levels to a significant extent. You can see a nice dose-dependent increase in Syngap protein in all these different brain regions, from frontal cortex, hippocampus, occipital cortex, and motor cortex. We also looked at some other regions, and we similarly saw increases in Syngap expression, upwards of around 1.5 to 1.7 fold. The mid-dose group was approximately the maximal effect, although in some cases there might be a little more at the high dose. But suffice to say, we're very excited to see that the ASO concentrations that we're able to achieve with the dosing intrathecally are able to increase Syngap in a dose-dependent manner. So if we look at our whole set of data that we've shown you today, we were able to identify this regulatory RNA that controls Syngap expression. So that sort of discovery, as Caleb referred to our special sauce, is really something that we were able to pioneer here. And then discover the oligonucleotide through screening through hundreds of oligos, finding those that had the most significant effect on Syngap levels and were well tolerated. And then we were able to demonstrate that they increased Syngap levels, restoring them towards wild type in patient iPSC-derived neurons. We showed also that they increased Syngap in a humanized mouse, and that this increase in Syngap was able to improve multiple phenotypes associated with Syngap haploinsufficiency. And finally, when we administer the oligonucleotide by IT administration, we're able to get enough ASO to the right places in the brain to increase Syngap to a significant extent. And I mentioned this during the NHP section, but importantly, the ASO has been very well tolerated in all the studies that we've run to date. And this is extremely important because we don't want to just increase Syngap, but we want to do so safely. And that's really the primary thing that we're interested in as we get into patients, is that the oligo will be safe. Now, the next step is to do formal GLP toxicity studies, and this is something that we'll be initiating later this year. GLP stands for Good Laboratory Practices, and this is are toxicity studies that are very highly regulated and required by regulatory agencies before you can go into human studies. So with that, I'd like to pass it on to Yuri to wrap it up.
Great, thanks so much, Dan. So as Dan mentioned, what we would often describe in the clinical or scientific terms of very robust data showing increase in protein and also improvement across a number of the aspects or domains of Syngap. I think I would use the human terms of impressive data showing the improvement in increase in protein as well as improvement in features of Syngap. And so the next steps, as Dan outlined for us, are to initiate the GLP toxicology studies. We are also actively engaged in continuing to work very closely with SRS, as well as scientific and clinical experts to prepare for future potential clinical trials within Syngap. And this also includes putting together the data packages, which includes the safety data as well as the data showing benefit in animals for regulatory authorities and starting to work on preparing for potential clinical studies across multiple regions of the world. So with that, really appreciate your time and attention. Happy to turn it over to you, SRF, for Q&A.
Thank you, everybody. Katrien, if you like, I've been collecting the questions so I can start putting them across to the team. First, I'm going to present myself. Hi, I'm Jaime Aranda. My son, Kai, 6-year-old, he's Syngapian, and I'm collaborating with Katrien in SRF EU. So we have received quite a few questions, at least 15. Many of them, I think they have been already answered by just, I'll just put them through and see if you can answer. So the first one is what are the inclusion criteria for the trial? I think we have answered this. The trial, the criteria is not determined yet, and it's one of the questions that we are not able to answer at this point due to the phase in which the preparation of the trial is. Is that correct, Yuri?
Jamie, maybe one thing I could just say here. So absolutely, we are working on what that might eventually be in the eventuality that we run a trial in Syngap. However, I think I can provide some general guidance from other trials, and the slides that Katrien walked through at the beginning are really helpful. So typically, the first in human studies, those are phase 1 or phase 1/2, in general have more limited criteria on who is appropriate, whereas oftentimes later studies tend to have slightly more open criteria. So in general, I think one thing as we go through this journey together is, as we all get more data, is to think about what stage of trial might be most appropriate for your family member if you think a trial is appropriate. And as we have more information, we will obviously share it with you all.
And on top of that, there is a question in relation to families being able to participate in different phases. So as in, if one participate in phase 1, 2, can they also participate in 2 and 3 or phase 3?
Yeah, thank you so much for this question. So in general, if an individual participates in one stage, they typically are not permitted to participate in another stage. And that's because we want to have overall general patient counts. This is typically a requirement by regulators. However, I want to highlight something that Katrien mentioned, which is quite common in rare disease clinical trials, which is this idea of an open label or expanded access. So we understand in devastating and significant diseases like Syngap that there are two things. One, people who might have their child enrolled in a earlier stage study would want to continue to receive that if we continue to see safe data. And so these open label expanded access could allow two things. One, they would allow those individuals who participate and received potentially the study medicine early on to continue to get it later at the end of their phase 1/2. They can roll into that. So you can almost think of it as a separate study that it is allowed for people to pull into. But the other thing is, if there is a placebo in the study, it also allows those individuals who receive the placebo to also roll into it. And in a condition like Syngap, it actually has an additional added benefit because it generates additional safety data where we can go and say, look, the number of patient years is even higher, so we can share that with the regulators. The second thing is it actually allows us to continue to track longer term outcomes that might not show up in shorter trials. And this is really important, I think, in a condition like in Syngap, because in Syngap, there might be some outcomes that we are able to observe a difference earlier, but there might be other outcomes that take much longer time. And so these open label expanded access is a great way for us to continue to monitor for those.
Okay. Somebody's asking right now, like, what is the placebo? And I think that question came out yesterday as well.
Yeah.
Like, can you explain?
So again, I'm not going to speak specifically to our clinical trial because those details haven't been fully worked out, and timing has not been fully worked out, etc. But for other studies where ASOs are used and injected intrathecally, and there's a placebo, typically a synthetic CSF. So it's basically a fluid that is sterile, so it's safe to be given, and it also is similar in terms of the volume, so that the safety is established.
Okay. The next question that I would like to put across is, would children who have abnormal EEGs but no clinical seizures could be considered for the trial?
Yeah, so this is, I think, a criteria question. And so at this stage, we're not able to provide any particular guidance to what those inclusion, exclusion criteria or study criteria are. I do think, though, again, I would point to the fact that oftentimes there may be different populations who may be most appropriate for different stages of trials. And sometimes there may be special populations, either by age or features of the disease, that may end up being a separate trial. And so as time goes on, there will be an opportunity to discuss those aspects further.
Okay.
I actually have a question here on sites, maybe, Yuri, if I can jump in.
Sure.
How are you going about choosing your sites? And if a site is not in someone's local country, can they travel to a different site, and will travel be facilitated?
Yes, so this is an important question. One is that I think in general in most studies, because the earlier phase 1/2 compared to the later phase 2 or phase 3, the earlier ones tend to be smaller. There tends to be a fewer number of sites, whereas we get into later stage studies, there tends to be more sites. And so it's a likelihood that there's more sites that might be closer to an individual's home. However, it is commonplace, and I would expect in a potential situation of a Syngap clinical trial where the travel of the patient and reasonable number of family members is supported. And that is commonplace because it is really important that we work to make sure that those who are interested in participating in a clinical trial and qualify are able to participate in that trial, and that financial resources is not a prevention from doing so. I think the other thing I'll just add is there is continued work, and I personally have done a lot of work on how do we enable clinical trials that maximize the amount of activity that can be done at home or nearby. So not all aspects of the clinical trial always has to be conducted at the specific site. There may be lab draws, there may be other kinds of observations or even visits that can be facilitated at home or nearby home. And I think that's really important given that participating in clinical trials is a lot of work. And so we want to maximize how we can make that as convenient as possible for everyone.
We have questions about the duration, normal. What is common for the duration of the different phases of the trial? So in general, we know that you guys are preparing the clinical trials, but like in general, people have questions. Would the phases be?
Yeah, it's really dependent on the disease. So different diseases have different phases. I can't speak specifically to what the potential periods might be in a future potential Syngap trial. I will say also too that in general there's what's called the treatment period. So that's a period of how many doses and how long people are getting dosed. Those tend to be shorter in the earlier phase and longer in general in the later phase. But the specifics is going to depend on a specific trial, specific type of medicine, and specific condition. And that's something that we can talk more about in the future.
Perfect. Then we have a question. Are any other ASOs working with displacing the restrictor?
Dan, do you want to speak to that?
Yeah, I mean, so I think, what we're getting at are there other ASOs that are targeting the regulatory RNA or different ways of increasing Syngap? We typically are screening the entire regulatory RNA and we get oligonucleotides that are increasing expression, and we really hone in on those that have the biggest effect. But there always will be multiple hits or multiple ASOs of slightly different sequence that have an effect. So there are others, but we went through a lot of work to figure out what's the best ASO, both in terms of the sequence, the effect on Syngap itself, as well as safety and tolerability of the ASO. So.
I have a question. If there were any noted side effects in the monkeys when you gave them the drug.
Right. So this is something that we really pay a lot of attention to. Even before we got to the monkey study, we had done a lot of mouse studies and confirmed that when we administered them into the ventricle within the brain, that there were no side effects of note, because we really want to make sure we're selecting for the most well-tolerated oligonucleotides. So when we went into the monkey study, this was something that we paid attention to. Again, this was a pharmacology study, meaning we wanted to look at the ASO concentration in Syngap, but we also wanted to get any information about how the ASO was tolerated by the monkeys. There were no signs or symptoms, meaning when we administered the ASO or the artificial CSF, the vehicle control, there were no differences in a response. So the monkeys did not experience any side effects at the dose levels and dose frequency that we administered. Now, of course, the next steps in our program to do these toxicity studies, that's where this will be formally addressed in a very rigorous and regimented way moving forward.
So as a follow-up on that, it was a question about overdosing in terms of how to get the dosage right and what happens if you go above the 100% Syngap1 level, how in humans, how's that going to be measured, or how's dosage going to be determined?
Yeah, that's great. Just a great question. I'll start at sort of the mechanistic level. What we have found are oligonucleotides that target these regulatory RNAs. The impact that they have on gene expression is within what we call a physiological range, meaning around twofold increase. So we have not encountered a situation where we can get a 5 or 10-fold increase of a protein that would really could potentially be problematic. That's different from potentially other modalities where it might be more difficult to control the dosage of the gene that you're trying to express. But we really are only going to increase about twofold. So in the context of a haploinsufficient state, we're just going to get back up to wild type. Maybe we get a little above wild type, maybe we get a little bit below wild type. And I think all those, there's no data to suggest that being a little bit above wild type is going to be problematic. We will be assessing that in the monkey studies because those will be wild type animals that have 2 copies of Syngap. So we will be going beyond. So I think that will, the data from that study will also provide further confidence that the oligo is well tolerated and safe, and that there's not a risk of going above that. Now, in patients, it's difficult to measure, Syngap. It's not like we can, go into the brain and directly measure Syngap. Sometimes you're able to look at a protein in the CSF, and sometimes you're not. Not all proteins end up in the CSF. We're exploring this, but we might not necessarily have to measure that directly to understand the impact of oligonucleotide that we're having.
The other question that we had is if the drug is tested in younger patients, once it's approved, will it be approved for all ages? Will it be approved for specific ages? How is that usually sort of approached in trials?
And
Yeah,
in addition,
so
if I can piggyback on that question too, just so that you can give maybe like a more complete answer. We have other questions about the different in effect between providing the ASOs to a child versus a young adult, like a 20-year-old. Are you expecting what difference in results are you kind of like thinking that they will have?
Yeah, so I'll maybe kind of take those in 2 parts, but they're definitely related. So it is very important, and there's a lot of thought both around the safety data as well as potentially efficacy data for studies in rare diseases around the age. And oftentimes there's a specific plan that's put in place around what number of participants across all the different trials might be at different ages. And so this is oftentimes an active conversation with the regulators. Typically, in an ideal situation, you have a certain number of participants across those ranges that give regulators confidence that people across those ranges that the medicine is both safe and effective across those ranges. The other thing I think, just to mention in terms of adults, and Dan, feel free to embellish, is that there is data that has been published around the ability in adults where a insertion of the Syngap gene has shown rescue. And so I think in principle, this gives us confidence that there might be benefit across multiple ages. Dan, if you want to add anything to that.
Yeah, there was this paper that nicely demonstrated sort of genetic reintroduction of Syngap in older animals. So these were basically adults. So in adult mice that had Syngap haploinsufficiency phenotypes, if they introduced it in older mice, they were able to reverse a number of those behaviors in response to seizures and things like that. So I think that highlights, and while there is certainly a role for Syngap, early in development, and there's a role for Syngap in neuronal activity throughout life. So even introducing later. Now, again, that's animal studies, that's a mouse study, but I think, there's a lot of conservation in biology. So I think that's the best thing that we can point to for that.
I think we have to talk in this webinar about the logistics of it. There are some questions about who is funding this clinical trial, whether it's funded or not, or whether it will be self-funded. Would the families will have to incur any costs, or how, if you can talk about that a little bit?
Yeah, in the vast majority of sponsored studies by companies like CAMP4, so biotech companies, the sponsor, which is the biotech company, funds the entire study. So that includes the cost of the treatment, that includes the cost of the study visits, and as we talked about earlier, that also includes reasonable travel costs as well.
Perfect. I think there is an interesting question that just came in. Is if Syngap is found in other neurons in the body, could this be something that could be given to improve non-brain Syngap? Like I remember in the conference last year, there was a researcher that was they were looking into the effect of the Syngap protein in the intestines, the digestive system. So
Yeah, I can maybe start there. So, one thing that that Yuri was talking about early on about, intrathecal administration being, most direct and proximal to the target organ is definitely true, right? But, oligonucleotide also does leave the brain, it leaves the CSF over time, and you do get oligonucleotide throughout the body. That said, it is at lower concentrations typically because the highest concentration is being directly into the CSF. So we certainly have heard about this and thought about that. So it really just depends on how much of the oligonucleotide that's leaving the CSF, is it getting into those tissues, and is that at a high enough concentration to have an effect? I mean, that's something we haven't looked at to this point, but it is interesting to consider.
yeah. I'm worried about time. We are already 5 minutes past.
We have so many questions.
We have
Maybe
so many.
can there be one question I just want to jump in on, Jamie?
Yeah.
I think is the language question. I think this is really important for Europe, which is it is standard approach that for clinical trials being run in other countries where there are multiple languages, that all materials as well as the site investigators at those sites are supported across multiple languages. So I think that's really important to make sure that everyone understands that when we conduct clinical trials in sites or countries where there are multiple languages, that those are supported to the best we can.
I think there's a lot of questions, and I completely understand everyone, and it's great that everyone's asking all these questions. I think it just shows the level of enthusiasm and appreciation. Everyone wants just some more information. A lot of them, I think that would be difficult to answer sort of right now, whether you're accepting very specific variants. So things that I think we'll just have to come back to also sort of at a future date when there is more concrete information, I think, about the structure of a trial.
Yeah, I would like to say that I'm happy to collect all of the questions, and maybe offline we can work with CAMP4 and getting the answers, and we can put them in a newsletter, or when the webinar is uploaded, we will attach a document with all the answers.
And we'd be happy to work with you on that.
Thank you so much, everyone, for your time, for all of the questions. Thank you so much to the CAMP4 team. We really appreciate it. You have now done 3 webinars, 3 different geographies. I'm sure you're sort of bored of seeing these same slides, but for a lot of us, it's a lot of new information, and so thank you so much for helping break it down and explain it to our community.
Thank you.
It's very motivating for us, Katrien. We feel really humble and privileged to be with all of you. So
yeah, happy to talk to you. Thank you.
Yeah, thanks so much for having us, and we look forward to the next opportunity to speak with you as well.
Thank you so much.
Thank you.
Bye.
Thank you, everyone. Thanks.